India, CDSO, Ahmedabad. I request Shri Jayant Kumar, Dr. Preeti Mehta, Shri J. Ramnivas, and Shri Vineet Mehta to come upon the dais and take their chairs. Thank you, everyone. Now I request Mr. Jayesh Joshi to welcome Shri Jayant Kumar with a bunch of flowers and present him a memento. Sir has 15 years of experience in Central Drug Standard Control Organization, DGHS, Ministry of Health and Family Welfare, Government of India. He worked as drugs inspector in Central Drugs Control Organization, West Zone, Mumbai for 7 years. He has participated in various scientific group discussions and regulatory policies of revised schedule, bioequivalence, bioavailability studies, clinical trials, stem cells, QA/QC in vitro diagnostic kits, blood products, and guidelines on similar biologics. Thank you so much, sir. Now I request Mr. Yasdeep Singh Jadeja to welcome Dr. Preeti Mehta, Chair Professor, Institute of Pharmacy and Head Department of Pharmaceutical Analysis, Ahmedabad, with a bunch of flowers and memento. Ma'am has more than 23 years of teaching, research and industrial experience. She is pioneer in getting interdisciplinary research grants at Nirma University and has worked at agencies like Ayush, ISRO, BRNS, Gujcost, etc. Thank you so much, ma'am. Now I request Mr. Parth Sheth to welcome Shri J. Ramnivas, CEO, Sai Pharma Solutions, Vadodara, with bouquet and memento. Sir has led various positions including Vice President, President, and Director of Quality and Regulatory Affairs in pharmaceutical industries such as Lupin Limited, Zandu, Zydus Cadila Healthcare Limited, Tonera Pharma, etc. Thank you so much, sir. Now I request Mr. Anas Sheikh to welcome Shri Vineet Mehta, Advocate, Gujarat High Court, Ahmedabad, with bunch of flowers and memento. Sir is practicing advocate at High Court of Gujarat, Civil and Sessions Court, Consumer Court at Ahmedabad. He holds MCom in Advanced Accounting and Auditing, LLB, LLM in Constitutional Law and Legal Order, Diploma in Labor Laws and Practice, and Diploma in Tax Laws and Practice. Thank you so much, sir. Now I duly request the chairperson of this session, Shri Jayant Kumar, to take over from here. Please, sir.
Good afternoon, one and all. My dear friends, dignitaries on the dais, J. Ramnivas, Ji Dr. Preeti Mehta ma'am, and Advocate Shri Vineet Mehta. After lunch, we have gathered here and we have the heavy doses of all the sessions. Lot of information we have already gathered, and we are very fortunate to, after lunch, going to start session. So we are going to start without much formality and giving overview for the regulatory and scientific requirement to get approval of biosimilar product by Dr. Preeti Mehta ji. After that, change in regulatory environment of Indian pharmaceutical sector and emerging opportunity and challenges by Shri J. Ramnivas ji. And most important topic, intellectual property right in the pharma industry. So I have been requested that Shri Vineet Mehta have some important work and he has to attend court. As per organizing committee, I am requesting him to give his talk on the intellectual property right in the pharma industry. Thank you.
Good afternoon, everyone. Here we all gathered for the discussion regarding the pharmaceutical and IPR in a pharmaceutical. Actually, I discussed more about it, but due to some emergency work, I'll have to go to the court. So I'll just conclude within the 5 to 10 minutes. As you all know about the different aspects of the IPR, and my agenda is that the IPR and its attributes, then constitutional aspects of the IPR in India. Because as the frame of the constitution, the article 19 clause F define the right to property. But after the 42nd amendment and as far as in the 44th amendment of the constitution, right to property under article 19 clause F is repealed and it include under the article 300 and A. And article 300 and A is specifically talked about the tangible property. But ultimately, while the interpretation of the constitution by the safeguarding body of Supreme Court and the different high courts, they specifically mention the article 300 and A. It's also safeguarding the intellectual property rights. But unfortunately, the intellectual property rights and no specific definition in any statute in India. So ultimately, when the case are they go up to the court and they define the what is IPR and what are the different attributes over there. Next is TRIPS. As you all know about the agreement with the relating to the trade related aspects of IPR, then role of patent cooperation treaties, that is PCT, then intellectual property and its role in pharmaceutical industry, then some aspect of drug patent specification and pharmaceutical patents in India and the effective tool for the development of Indian pharmaceutical industry. As we all know about the different attributes, there are the few types of intellectual property. First is copyright related and the related rights, then trademarks, then the geographical indication, industrial design, layout design of the integrated circuits, then the trade secrets, patent pertaining of the microorganism and the new patent veracity. So these are the different. The very first case where the IPR is defined, the case the Institute of Chartered Accountant of India versus Shankal H. Satya and others. In that case, the court has specifically defined what is IPR. And still we are in that phase. The court immensely presents and immensely statute that there are the different types of IPR there. So there is a question, what is right, what is property and what is intellectual property? These are the different significance are there. If we are talking about the rights, then our constitution specifically defines the fundamental rights under the part 3 of the constitution, as we all know about it. But what is intellectual property rights? Whether it is covered under the fundamental rights, so answer is no. Constitution specifically told about that the intellectual property rights or any right to property under article 308 give the constitutional rights. So which is ultimately important for us. When the legal aspects are there, when the legal rights are there, then you have to approach the appropriate court. And for this IPR, we are talking about the IPR. So IPR you may go to the High Court and Supreme Court. But in same way, Supreme Court also observed that you are not directly come to the Supreme Court. You have to go first to the High Court and then come with us, that is Supreme Court. So there are specific mechanism and regulation are there, which ultimately we can say that we are stuck upon it. There is no specific resolution over it. If we are talking about the pharmaceutical industry only, then section 3 of the Patent Act 1970 is most important. In section 3, it also observed in a different case laws by the High Court, Delhi High Court, Bombay High Court, Supreme Court as well as, there is a ultimately discrimination upon the sections 3, clause D, clause E, clause I, clause F. So ultimately, I can say that the Supreme Court and High Court justified and interpret the IPR in a different manner in a different cases. No doubt cases are cases, the judgments are derived and different, but there are some substance. There are some basic limitations over it, so we have to accurately it. If we are talking about that the IPR, so there are trademark also, patent also, copyright also, it all has the own jurisdiction or own statute, and all statute define different things. But ultimately we are going to the correction over it, on the conspirate over there, or collect at a one place, then ultimately you have to safeguard your right as per the Constitution of India. So I can suggest or I can told that ultimately the constitution is important whether the statute is defined or not defined. Before 1970 there is no question of the IPR. If we are going to the history, then the ancient history for the India, ultimately we approach that what is IPR, we define what is IPR. If I am talking about the mythological perspective, so ultimately the inventions are created in India only. Then after the world goes on and world talk about these different things. Next topic is that is a TRIPS, that is trade related agreement to the IPR. There are the different agreements. We are also the member of the WTO, so we have to follow that regulation also. Then we are also with the WIPO also, the member of WIPO also. So we have to over it and we have to strike down or we have to regulate according to them. So there is a questionable thing whether we are approaching some different thing and they are or different companies are shoot against him. So there are this few discrimination over it where the IPR is actually getting into touch or not. The one case I found it, that is the IPR is, there is a case relating to the patent. The patent in a different form. In a pharmaceutical industry, there are 6 types of patents are there. So ultimately you have to follow that 6 particularly patent registration. The one company follows that, whether that company, the suit is going on in the High Court. At that point of time, another company can use that patent. There is a question mark. Because according to the Civil Procedure Code, the different apologies are there, the different remedies are there, different damages are there. So as per the Supreme Court guideline, wherever the suit are going into the court and suit are going on the parameters, at that point of time you cannot do anything with respect to that case. But in India, there are some loopholes, there are some regulations, so ultimately they are going into it. So there are the different challenges over it also with respect to IPR, and there are, we can say, the favorable goods thing also. The one of the good thing and bad thing is that the impact of IPR, that is a competition from the MNC, and litigation work in India is, to be very honest, very costly. As per not, I am advocate, that's why I am telling you, but it's a very costly in a nature. क्योंकि यहाँ इंडिया में है ना, एक्चुअली बताऊँ ना, एडवोकेट्स की हालत वैसे भी बहुत अच्छी है, नो डाउट, बट लिटिगेशन कॉस्ट सच में ज़्यादा है इंडिया में. क्योंकि अगर कोई इंसान हमारे घर में मर भी जाता है ना और उसकी प्रॉपर्टी है ना, तो उसके रिगार्डिंग भी हमें खर्चा करना पड़ता है. इंडिया की हालत ये है. और अगर यही हालत मैं यूएसए की और से कंपेयर करूँ ना, तो उनका कॉन्स्टिट्यूशन इतना ईजी है कि कोई भी गुजर जाएगा ना तो भी वो ईजीली अवेल इट. उनके पास सारा डेटा होता है, वो ही खुद करके देगा कि भाई नहीं, इनके ये पुत्र है, इनकी ये पुत्री है और ये काम हो जाएगा. So as far as concerned with the India, it is ultimately litigation cost is a very high in a nature. Price rises and good for sign, that is the growth of the pharma industry, then more resources would be devoted in the basic research, and global research industry and outsources are very high in a nature. So in nutshell, I am telling about you with the IPR. We have at that stage we have to improve ourselves. If we are talking about the only pharmaceutical industry, then as per my research, when I was in a master's in commerce and I was do the research with respect to the working capital of pharmaceutical industry, at that point of time few companies are in a good manner. So as per that research and as per concluded with today's session, 2047 may be good for the India in a pharmaceutical industry with respect to IPR. But if Indian governments go through the act and going to the very mechanism in a very easy manner, so we will achieve the 2047 goal. So these are the different IPRs are there. So So I'm concluding it today because there are some emergency. Sorry, but thank you. Okay. आपको
Thank-
पीपीटी मिल
thank-
जाएगी क्योंकि इसमें मैंने सारा रिसर्च किया हुआ है एज वेल एज 10 लैंडमार्क जजमेंट्स भी मैंने दिए हुए हैं. अगर कोई भी इंडस्ट्री के रिगार्डिंग हो, आपके वहाँ पे एडवोकेट्स तो हो गए डेफिनेटली, बट दिस जजमेंट्स आर टॉकिंग अबाउट मेजरली फॉर द पेटेंट एनालिसिस ओनली. और इंडिया के अंदर ये बहुत ही इम्पोर्टेंट है. और ये लैंडमार्क जजमेंट्स ऐसे है कि कई बार इंडियन गवर्नमेंट्स और इंडियन कोर्ट्स में इंग्लैंड से भी ये केसेस के जजमेंट्स के रेफरेंस दिए हुए हैं. सो जहाँ पे इंडियन गवर्नमेंट आईपीआर में क्वेश्चन मार्क हो जाता है, वहाँ पे वो इजीली यूएस या तो इंग्लैंड से ये केसेस लेते हैं और वहाँ से वो जजमेंट उसके बेसिस पे इंटरप्रिटेशन करके यहाँ पे हमें ये जजमेंट्स मिल जाते हैं. सो यू कैन अवेल इट. सो इट विल वेरी मच हेल्प यू. ओके, थैंक यू.
Thank you, Vineet Mehta, for your short and summarized lecture, and very nice about the patent life. Any question, because he is leaving? So, Asha, 1 minute, 1 minute. Any question? He is leaving. Otherwise, you permit me. I will also, because he is in very. Okay, okay. Okay, no problem, no problem. So he has nicely, Vineet Mehta has about the IPR and Section 3 of 1970. And in this audience would like to hear about something compulsory licensing, what are the impact of that, but he might not have time. So he has given his presentation, we will share it. And now we are moving to the second speaker, Dr. Preeti Mehta, and she will deliver her valuable speech, and she will enlighten about the regulatory and scientific requirement to get approval of the biosimilar product in India. Thank you.
Good afternoon, everyone. First of all, thank you to the organizers for providing this platform to interact with all of you. And I think it is a very difficult task for me because this session is post-lunch. So it is another responsibility on me to keep you all awake, isn't it? But hope I will meet your expectations. Today I choose the topic on regulatory and scientific requirement to get approval of similar biologics in India. As a country, we have proven ourselves as a generic manufacturer in small molecules, as well as we are also shown our potential in the vaccine development, as well as we are providing the vaccines to many countries all over the world. And now this is the segment which is calling us that yes, you can prove yourself over here also. So little bit glimpses I'm giving over here, what I know, and this is my views, as well as I try to, this presentation will mainly include scientifically how the biologics are different than the small molecule, and secondly, how this regulated in India and what further need to be updated in the regulations of such type of molecules. When we talk about biologics, there are a number of categories in biologics. It is vaccines, it is plasma-derived proteins, toxins. I am mainly looking for recombinant therapeutic proteins and monoclonal antibodies. They are basically known as biopharmaceuticals, and normally we use microbial platform or cell lines to develop such type of product. Now, what is the impact of COVID-19 on the development of such type of products? Many biopharmaceuticals developed and tried to be used for the management of COVID. Many countries came forward for that. So definitely COVID-19 has a positive impact on the development of biotherapeutics. we know that along with the remdesivir, tocilizumab was also recommended by the government authority to be used for the patients who are hospitalized and for the management of the cytochrome storm. But it was not much available because only one company all over the world is providing tocilizumab, and the no biosimilars are available till that time. Along with it, number of monoclonal other antibodies are also under clinical trials for the COVID management. Along with that, few cytokines, which are interleukins, as well as number of peptides also developed for the management of the respiratory syndromes, and the clinical trials are still going on. Some are repurposed also. They try to see the other indications also for the peptides or proteins which are developed during that time. So, little bit about the difference between the small molecule and large molecule. I know this is the learned audience and know very well that small molecules, chemical molecules, and large molecule, which can also segregated into peptides, growth hormones, which are a medium-sized molecule, as well as monoclonal antibodies, a very, very complex structure it is having. And if we see the growth rate of the biopharmaceutical industry in India, it is projected very well with the growth of 9.1%. And segment-wise also, the monoclonal antibodies and peptides have a very huge market or a very huge area where one can develop the expertise and have a good business opportunities. Why as a India we can think over it? The reason is, again, generic comes in the mind, that many patents, this is the second patent cliff, where many products is going off patent, and it is available to develop the generic formulation of it. We cannot say that generic for biotherapeutics, but similar formulation of it. India is doing well. We are not very, we are not very behind. We are doing well. Many biotech companies are there, but still we have to improvise. The one positive sign I would like to say is our regulations, Indian regulations are at place. With whatever the, we are not going in the intricacies, but today maximum number of biosimilars are approved in the world by Indian regulators. That means in India, many biosimilars are approved because of the proactive stand of the regulators in India. So this is basically to give affordable medicines to the population of our country. Now, what the complexities involved in it? It is like if we would like to do the characterization and if we would like to handle the small molecule, then it is like riding a bicycle. But if you would like to handle a large molecule like monoclonal antibodies, it is like having expertise to fly jet plane. So you can understand the complexity involved, or you can understand the type of the expertise required when we would like to handle such a large molecule protein. But it is not too difficult if we are working as a team. We know that bicycle we can ride alone, but to fly a jet plane successfully, a team of people can work it, and we can do the task easily. So same thing is also applicable when we would like to do manufacturing of large molecules like monoclonal antibodies or peptides. Now, first of all, what do you mean by such biotherapeutic products? So, how it is developed? Basically, they are from the recombinant therapeutic proteins, and they are derived from the living sources like culture bacteria, yeast, cells, or we can use human and animal cell lines for the development of such product. So this is totally different than what we are doing for the small molecules, where we are doing the synthesis. API we are synthesizing in lab. Here, for the synthesis of API of biotherapeutics, we need living organisms. We need that type of fermentation processes. My talk will not go in detail about the novel biotherapeutics, but I'm talking about the biosimilars, which are more like a generic version of the biotherapeutic product. So in US and Europe, they considered a word biosimilar, and for that they say that the developed formulation or the product should be highly similar, or they have a highly similar active drug substance, and one has to prove that there is no clinical meaningful difference in safety, efficacy and purity of the product. CDESCO used the word, in India we are using the word similar biologics. So different agencies are using different word, like WHO is talking about similar biotherapeutic product, Europe biosimilar, and we similar biologics. So in a very brief how it is developed. As we need microorganisms, it is grown, cultured, grown, and then it is expressed. So the protein molecules get expressed, which we collect, purify it, and we make it into the formulation. So the manufacturing setup or which required is like a gene editing or gene cloning, which is further host cell system we need, in which such type of genes can be expressed, which can give maximum proteins generated by the cell. Then we have to culture such type of cell using a fermentation techniques, purification is required, and then finally the product is out. And mostly biotherapeutics products are IV based products, so we are giving using they are the injectables. Now, how the regulators look at it? You develop the formulation, but how it is looked by the regulators? So when we talk about the novel biologics, then more emphasis is given to clinical trials. But when we are talking about biosimilar products, then more emphasis is given to analytical characterization and functional characterization. So if we are developing such formulation, then if we match it or if we prove that analytically there is no difference in the product, functionally also both the products are same, then there is little need to be done on clinical studies. We all know that to do the clinical studies, huge money is required, isn't it? And which make the whole project very costly. But when the clinical studies are less, then the cost of the product also get reduced. This is the basic funda, but at the same time, it is very, very essential to do the similarity assessment of the developed product with the innovative product, both the way, physicochemically as well as biologically. So the thought process of the regulatory authority goes with this, that if similarity is developed at physical and biological level, then chances of having similarity in clinical studies is very, very high. So there is no, if the similarity is established very well at analytical level or the formulation or the molecule part as well as the biological activity part, then chances of having this similarity between the innovative product and biosimilar product at clinical level is very, very high. So to demonstrate the similarity, one has to do the structural similarity, one has to assess the structural similarity as well as functional similarity, as well as to check or to demonstrate there is no clinical meaningful difference between both the products. Pharmacokinetic, pharmacodynamic studies is required, as well as it is very, very mandatory to do the immunological studies. And for this purpose, it is important to use the orthogonal approach for the characterization of biotherapeutics. As the molecule is very big, protein in nature, so much of folding is there, though amino acids are only present, but it is folded in a way that we have to determine the primary, secondary and tertiary structure of the protein, otherwise activity can get lost. If the primary structure is same for innovative product and for developed product, if primary structure is same but the folding, that means the tertiary structure is different, then also it can be clinically it is not giving the response. So the structural similarity to be established all three levels, as well as impurity profiling, purity of the molecule, everything to be determined by number of ways. So different techniques are required to do all this analysis. But it has a very good entrepreneurship potential. The reason, again, MSME is a very strong foundation of Indian pharma sector. And if we see the cost of such type of biotherapeutics is very high if we compare it with the average income of the person in India. So to make such type of products available, if the pharmaceutical sector is working on it, then huge potential is there to have a growth in this segment. Now if we see the role of MSME, as we have earlier discussed, that so much of small molecule production is, MSME's role is very high in supplying medicines in India, small molecule drugs specifically. Now to enter into this segment by MSME, the three major gaps need to be fulfilled. One is the instrumentation facility, which is very, very essential because such type of characterization is required, and for that type of instrumentation facility is required. So very huge investment is there. So such type of, as government is also coming up with the scheme where 5 or 6 companies come together, can establish to have common facility, which will be very, very useful if somebody would like to go in this segment. Second, lack of expertise. Yes, human resource development is very, very essential. Nowadays, people who are available, who are trained in the biotechnology area, but might be they are not much trained from the pharmaceutical or drug aspects. So such type of balancing of the human resource development is very, very essential to escalate the growth in this segment. Third one is the regulatory compliance. Knowledge about how to get the regulatory compliance of such type of product is also very, very essential, and that awareness also helps to work in this area very easily. So which are the different authorities involved for the approval of biosimilars in India? First one comes the Institutional Biosafety Committee, IBSC, which mainly constituted at the particular organization, and they look at the handling and production activities related to the genetically modified organisms or the gene editing and everything. And then they do the communication with the RCGM. RCGM is the Review Committee on Genetic Manipulation. This body is specifically involved to give the approval for the product development as well as for the preclinical studies. So this body give the approval. First of all, protocol need to be approved by this committee to do the preclinical studies, and then finally after doing the studies also one has to take the approval that the study is okay, and after that only one has to go for the clinical trials with CDESCO. Along with that, Genetic Engineering Appraisal Committee, GESC, is also there. When there is a living organisms are involved in the process, one has to take the approval of that committee also. So regulatory approval is quite diversified in this segment, and zonal CDESCO is responsible for import of the drug for examination, test and analysis for research and development, etc. So normally the reference product need to be import, and for that the zonal office can come into the picture. So basically guidelines of similar biologics is published by Department of Biotechnology along with CDESCO, which is used along with the guideline document given by National Institute of Biologics, and they are also involved in the approval of the products. ICH guidelines Q5CE and Q6B are the guidelines to develop, monitor and stability studies of such biotherapeutics product. So this is the glimpses of different types of forms required to get the approval for biotherapeutics product. Now little bit going in, when one develop the biotherapeutics or biosimilar products, the first thing is to do characterization of reference product. Which reference product you would like to take, and based on that product you would like to develop your product and would like to assess the similarity. So that selection of reference product is very, very important step. Along with that, identification of critical attributes as well as key quality attributes. These are very important step to do. So whenever somebody selects reference product, it should be licensed or approved in India or by the ICH country. I think you are aware which are the different ICH countries, and at least product should be licensed there or approved by them, then you can take it as a reference product. And you cannot take any other similar biologics or biosimilar product which is already available in the market as a reference product. Always you have to take the innovator product for your comparison purpose. When we talk about the monoclonal antibodies, then dosage form, strength, route of administration should be same like the innovator product. But when you are talking about the peptides, little bit relaxation is there in that case. So first, to do the analytical comparison, as I mentioned earlier, you have to prove the similarity by number of ways. You can primary structure, secondary structure, tertiary structure, even which type of impurities get generated in your product and which is already generated in the reference product, it should also to be matched. And you need a different characterization tools like LC-Q-TOF, HPLC with fluorescence, DSC, circular dichroism, SDS page. Normally we are thinking of only HPLCs in pharmaceutical industries, but when we talk about the biotherapeutics, we have to think of all other analytical instruments also. And for biological characterization like surface plasmon resonance spectroscopy, because they are basically these instruments are used to check the binding studies as well as the biological effectiveness of the developed product. Now after doing the similarity at analytical and biological level, one has to go for the preclinical studies. Sorry, might be these fonts are little bit very small, but what I would like to say is RCGM is the committee which can approve the protocol to do the preclinical studies. So before doing animal studies, you have to give the product development data. You have to submit the product development data to RCGM. Based on the data, physicochemical characterization and biological characterization data, stability data, they will decide whether to give the permission to start the preclinical studies or not. And once they give the permission, you have to, after conducting the preclinical studies, again you have to give the study reports and you have to take the approval. After that, it is important to start the clinical trial where CDESCO come into the picture. And PKPD studies are normally carried out in first phase of the clinical trials. So first phase of clinical trials is carried out extensively to check the PKPD for the developed product. Many a times, phase 3 clinical trial can be waived, but it is in India, it is going on case-to-case basis, as we don't have any product-specific guidelines. So depending on the data submitted and depending on the confidence in the data, the decision is taken. Apart from the PK/PD studies, it is very important to do the immunogenicity studies for any biotherapeutic molecule. Either it can be peptide or it can be protein. And one has to prove there is no meaningful difference in immune response generated by the proposed biotherapeutic product and the reference product. Now, we have many regulations at place in India, but recently the developed countries, USA and EMA, came up with the different regulations for the synthetic peptide. If here again I would like to draw your attention, when we are talking about monoclonal antibodies, they are very large molecules having amino acids in quite good numbers. But when we are talking about the peptides, which is around less than 40 amino acid chains, and such type of molecules can be synthesized by recombinant DNA technology like monoclonal antibodies, but at the same time, it is synthesized in lab also. So, peptides are of two types. One is synthetic origin and another one is recombinant DNA origin. And the regulations of synthetic origin peptides are taken with the route of small molecule approval by US FDA and EMA along with required immunogenicity data. But this clarity is not there in Indian regulations, which can be, I'm sure that it will come very fast, but this is there. So, you have, being a manufacturer, it is a, very dynamic and tricky decision to take. For an example, if synthetic peptide is developed, then how to get the approval of that molecule. But in 2020, the guidance is there by the CDESCO regarding how to get the approval of synthetically manufactured drug, which have been previously approved as recombinant DNA derived drug. So here, the case is one peptide which is recombinant, which is developed using recombinant DNA technology, okay, and it is approved already. Now you would like to develop another molecule using synthesis, okay, and you would like to get the approval. So here you have to compare the product which is developed using recombinant DNA technology and product which is developed in lab. So that comparison you have to do, and you have to take the approval as a drug. So if we talk about US FDA, they are taking a route of generic ANDA and product-specific guidance is also available. But if the peptide is recombinant-based product, then it goes with BLA or biosimilar guideline or regulations. So which are the gray areas where as a country we have to work? One is the introducing product-specific guidelines for biologic products like insulin, guidance note on biosimilarity as well as interchangeability. Many products, large molecule products are used for the other clinical indications, so in which case it can be used. So that type of guidance is also required to be at place. And yes, of course, innovative science-based approach is required to be taken further for the evaluation of such type of product dossiers. Thank you for your patience.